In biology, histones are highly alkaline proteins found in eukaryotic cell nuclei that package and order the DNA into structural units called nucleosomes. They are the chief protein components of chromatin, acting as spools around which DNA winds, and play a role in gene regulation. Without histones, the unwound DNA in chromosomes would be very long. For example, each human cell has about 1.8 meters of DNA, but wound on the histones it has about 90 micrometers of chromatin, which, when duplicated and condensed during mitosis, result in about 120 micrometers of chromosomes. Classes: 5 major families of histones exist: H1 per hour 5, H2A, H2B, H3 and H4. Histones H2A, H2B, H3 and H4 are known as the core histones, while histones H1 and H5 are known as the linker histones. 2 of each of the core histones assemble to form one octameric nucleosome core, approximately 63 angstroms in diameter-like particle. 147 base pairs of DNA wrap around this core particle 1.65 times in a left-handed superhelical turn to give a particle of around 100 angstroms across. The linker histone H1 binds the nucleosome at the entry and exit sites of the DNA, thus locking the DNA into place and allowing the formation of higher-order structure. The most basic such formation is the 10-NM fiber or beads on a string conformation. This involves the wrapping of DNA around nucleosomes with approximately 50 base pairs of DNA separating each pair of nucleosomes. Higher-order structures include the 30-NM fiber and 100-NM fiber, these being the structures found in normal cells. During mitosis and meiosis, the condensed chromosomes are assembled through interactions between nucleosomes and other regulatory proteins. The following is a list of human histone proteins. Structure: the nucleosome core is formed of 2 H2A H2B dimers and a H3H4 tetramer, forming 2 nearly symmetrical halves by tertiary structure. The H2A H2B dimers and H3H4 tetramer also show pseudodiad symmetry. The 4 core histones are relatively similar in structure and are highly conserved through evolution, all featuring a helix-turn-helix-turn-helix motif. They also share the feature of long tails on one end of the amino acid structure, this being the location of post-translational modification. It has been proposed that histone proteins are evolutionarily related to the helical part of the extended AAA plus AT base domain, the C domain, and to the N-terminal substrate recognition domain of CLPHSP100 proteins. Despite the differences in their topology, these 3 folds share a homologous helix-strand-helix motif. Using an electron paramagnetic resonance spin labeling technique, British researchers measured the distances between the spools around which eukaryotic cells wind their DNA. They determined the spacings range from 59 to 70 A. In all, histones make 5 types of interactions with DNA: helix dipoles from alpha helices in H2B, H3, and H4 cause a net positive charge to accumulate at the point of interaction with negatively charged phosphate groups on DNA, hydrogen bonds between the DNA backbone and the amide group on the main chain of histone proteins, nonpolar interactions between the histone and deoxyribose sugars on DNA, salt bridges and hydrogen bonds between side chains of basic amino acids and phosphate oxygens on DNA, non-specific minor groove insertions of the H3 and H2B. N-terminal tails into 2 minor grooves each on the DNA molecule. The highly basic nature of histones, aside from facilitating DNA-histone interactions, contributes to their water solubility. Histones are subject to post-translational modification by enzymes primarily on their N-terminal tails, but also in their globular domains. Such modifications include methylation, citrullination, acetylation, phosphorylation, sumoylation, ubiquitination, and ADP ribosylation. This affects their function of gene regulation. In general, genes that are active have less bound histone, while inactive genes are highly associated with histones during interphase. It also appears that the structure of histones has been evolutionarily conserved, as any deleterious mutations would be severely maladaptive. All histones have a highly positively charged N-terminus with many lysine and arginine residues. History: histones were discovered in 1884 by Albrecht Kossel. The word histone dates from the late 19th century and is from the German histon, of uncertain origin, perhaps from Greek histoni or from histos. Until the early 1990s, histones were dismissed by most as inert packing material for eukaryotic nuclear DNA, based in part on the ball and stick models of Mark Vtashen and others who believed that transcription was activated by protein DNA and protein-protein interactions on largely naked DNA templates, as is the case in bacteria. During the 1980s, work by Michael Grunstein demonstrated that eukaryotic histones repress gene transcription, and that the function of transcriptional activators is to overcome this repression. It is now known that histones play both positive and negative roles in gene expression, forming the basis of the histone code. The work of Vincent Olfrey on histone modification was a pioneering one. He is regarded as father of epigenetics. The discovery of the H5 histone appears to date back to 1970s, and in classification it has been grouped with H1. Conservation across species: histones are found in the nuclei of eukaryotic cells, and in certain archaea, namely the moproteals and euarchaea, but not in bacteria. The unicellular algae known as dinoflagellates are the only eukaryotes that are known to completely lack histones. Archaeal histones may well resemble the evolutionary precursors to eukaryotic histones. Histone proteins are among the most highly conserved proteins in eukaryotes, emphasizing their important role in the biology of the nucleus. In contrast, mature sperm cells largely use protamines to package their genomic DNA, most likely because this allows them to achieve an even higher packaging ratio. Core histones are highly conserved proteins. That is, there are very few differences among the amino acid sequences of the histone proteins of different species. Linker histone usually has more than one form within a species and is also less conserved than the core histones. There are some variant forms in some of the major classes. They share amino acid sequence homology and core structural similarity to a specific class of major histones but also have their own feature that is distinct from the major histones. These minor histones usually carry out specific functions of the chromatin metabolism. For example, histone H3 like CENPA is associated with only the centromere region of the chromosome. Histone H2A variant H2AZ is associated with the promoters of actively transcribed genes and also involved in the prevention of the spread of silent heterochromatin. Furthermore, H2AZ has roles in chromatin for genome stability. Another H2A variant H2AX binds to the DNA with double-strand breaks and marks the region undergoing DNA repair. Histone H3.3 is associated with the body of actively transcribed genes. Function: compacting DNA strands, histones act as spools around which DNA winds. This enables the compaction necessary to fit the large genomes of eukaryotes inside cell nuclei. The compacted molecule is 40,000 times shorter than an unpacked molecule. Chromatin regulation: histones undergo post-translational modifications that alter their interaction with DNA and nucleoproteins. The H3 and H4 histones have long tails protruding from the nucleosome, which can be covalently modified at several places. Modifications of the tail include methylation, acetylation, phosphorylation, ubiquitination, sumoylation, citrullination, and ADP ribosylation. The core of the histones H2A, H2B, and H3 can also be modified. Combinations of modifications are thought to constitute a code, the so-called histone code. Histone modifications act in diverse biological processes such as gene regulation, DNA repair, chromosome condensation and spermatogenesis. The common nomenclature of histone modifications is, the name of the histone, the single-letter amino acid abbreviation and the amino acid position in the protein, the type of modification, the number of modifications, so H3K4Me1 denotes the monomethylation of the fourth residue from the start of the H3 protein. Examples of histone modifications in transcription regulation include Functions of histone modifications. A huge catalogue of histone modifications have been described, but a functional understanding of most is still lacking. Collectively, it is thought that histone modifications may underlie a histone code, whereby combinations of histone modifications have specific meanings. However, most functional data concerns individual prominent histone modifications that are biochemically amenable to detailed study. Chemistry of histone modifications. Lysine methylation. The addition of 1, 2 or 3 methyl groups to lysine has little effect on the chemistry of the histone. Methylation leaves the charge of the lysine intact and adds a minimal number of atoms, so steric interactions are mostly unaffected. However, proteins containing TUDA, chromo or PhD domains, amongst others, can recognize lysine methylation with exquisite sensitivity and differentiate mono, D and trimethyl lysine, to the extent that, for some lysines mono, D and trimethylation appear to have different meanings. Because of this, lysine methylation tends to be a very informative mark and dominates the known histone modification functions. Arginine methylation. What was said above of the chemistry of lysine methylation also applies to arginine methylation, and some protein dormants for example, TUDA dormants can be specific for methylarginine instead of methyl lysine. Arginine is known to be mono or demethylated, and methylation can be symmetric or asymmetric, potentially with different meanings. Lysine acetylation. Addition of an acetyl group has a major chemical effect on lysine as it neutralizes the positive charge. This reduces electrostatic attraction between the histone and the negatively charged DNA backbone, loosening the chromatin structure. Highly acetylated histones form more accessible chromatin and tend to be associated with active transcription. Lysine acetylation appears to be less precise in meaning than methylation, in that histone acetyl transferases tend to act on more than one lysine. Presumably this reflects the need to alter multiple lysines to have a significant effect on chromatin structure. Serine tyrosine phosphorylation. Addition of a negatively charged phosphate group can lead to major changes in protein structure, leading to the well-characterized role of phosphorylation in controlling protein function. It is not clear what structural implications histone phosphorylation has, but histone phosphorylation has clear functions as a post-translational modification, and binding domains such as BRCT have been characterized. Functions in transcription. Most well-studied histone modifications are involved in control of transcription. Actively transcribed genes. 2 histone modifications are particularly associated with active transcription: trimethylation of H3 lysine 4 at the promoter of active genes. H3K4 trimethylation is performed by the compass complex. Despite the conservation of this complex and histone modification from yeast to mammals, it is not entirely clear what role this modification plays. However, it is an excellent mark of active promoters, and the level of this histone modification at a gene near unregistered trademark S promoter is broadly correlated with transcriptional activity of the gene. The formation of this mark is tied to transcription in a rather convoluted manner. Early in transcription of a gene, RNA polymerase II undergoes a switch from initiating a unregistered trademark to a elongating a unregistered trademark, marked by a change in the phosphorylation states of the RNA polymerase II C-terminal domain. The same enzyme that phosphorylates the CTD also phosphorylates the Rad6 complex, which in turn adds a ubiquitin mark to H2BK123. H2BK123 occurs throughout transcribed regions, but this mark is required for compass to trimethylate H3K4 at promoters. Trimethylation of H3 lysine 36 in the body of active genes. H3K36 trimethylation is deposited by the methyltransferase SET2. This protein associates with elongating RNA polymerase II, and H3K36Me3 is indicative of actively transcribed genes. H3K36Me3 is recognized by the RPD3 histone destylase complex, which removes acetyl modifications from surrounding histones, increasing chromatin compaction and repressing spurious transcription. Increased chromatin compaction prevents transcription factors from accessing DNA, and reduces the likelihood of new transcription events being initiated within the body of the gene. This process therefore helps ensure that transcription is not interrupted. Repressed genes. 3 histone modifications are particularly associated with repressed genes: trimethylation of H3 lysine 27. This histone modification is deposited by the polycomb complex PRC2. It is a clear marker of gene repression, and is likely bound by other proteins to exert a repressive function. Another polycomb complex, PRC1, can bind H3K27Me3 and adds the histone modification H2AK119 which aids chromatin compaction. Based on this data, it appears that PRC1 is recruited through the action of PRC2, however, recent studies show that PRC1 is recruited to the same sites in the absence of PRC2. D and trimethylation of H3 lysine 9 hours 3K9Me2/3 is a well-characterized marker for heterochromatin, and is therefore strongly associated with gene repression. The formation of heterochromatin has been best studied in the yeast schizos of Caramyces pum, where it is initiated by recruitment of the RNA-induced transcriptional silencing complex to double-stranded RNAs produced from centromeric repeats. RITS recruits the CLR4 histone methyltransferase which deposits H3K9Me2/3. This process is called histone methylation. H3K9Me2/3 serves as a binding site for the recruitment of SWI6 which in turn recruits further repressive activities including histone modifiers such as histone deacetylases and histone methyltransferases. Trimethylation of H4 lysine 20. This modification is tightly associated with heterochromatin, although its functional importance remains unclear. This mark is placed by the Su4-20 hours methyltransferase, which is at least in part recruited by heterochromatin protein 1. Bivalent promoters. Analysis of histone modifications in embryonic stem cells revealed many gene promoters carrying both H3K4Me3 and H3K27Me3. In other words, these promoters display both activating and repressing marks simultaneously. This peculiar combination of modifications marks genes that are poised for transcription. They are not required in stem cells, but are rapidly required after differentiation into some lineages. Once the cell starts to differentiate, these bivalent promoters are resolved to either active or repressive states depending on the chosen lineage. Other functions. DNA damage. Marking sites of DNA damage is an important function for histone modifications. Phosphorylation of H2-AX at serine 139. Phosphorylated H2-AX is a marker for DNA double-strand breaks, and forms part of the response to DNA damage. H2-AX is phosphorylated early after detection of DNA double-strand break, and forms a domain extending many kilobases either side of the damage. Gamma H2-AX acts as a binding site for the protein MDC1, which in turn recruits key DNA repair proteins and as such, gamma H2-AX forms a vital part of the machinery that ensures genome stability. Acetylation of H3 lysine 56, H3K56ACX is required for genome stability. H3K56 is acetylated by the P300/RTT109 complex, but is rapidly deacetylated around sites of DNA damage. H3K56 acetylation is also required to stabilize stalled replication forks, preventing dangerous replication fork collapses. Although in general mammals make far greater use of histone modifications than microorganisms, a major role of H3K56 in DNA replication exists only in fungi, and this has become a target for antibiotic development. Chromosome condensation, phosphorylation of H3 at serine 10, the mitotic kinase Aurora B phosphorylates histone H3 at serine 10, triggering a cascade of changes that mediate mitotic chromosome condensation. Condensed chromosomes therefore stain very strongly for this mark, but H3S10 phosphorylation is also present at certain chromosome sites outside mitosis, for example in pericentric heterochromatin of cells during G2. H3S10 phosphorylation has also been linked to DNA damage caused by a loop formation at highly transcribed sites. phosphorylation H2B at serine 10 in yeast or serine 14 in mammalian cells. Phosphorylation of H2B at serine 10 or serine 14 is also linked to chromatin condensation, but for the very different purpose of mediating chromosome condensation during apoptosis. This mark is not simply a late-acting bystander in apoptosis, as yeast-carrying mutations of this residue are resistant to hydrogen peroxide-induced apoptotic cell death. See also. References. External links: chromatin, histones and cathepsin. PMAP, the proteolysis map animation.